These are the lecture notes for the Chapter 25 slides, Part B, and we are actually going to skip through this first little bit of information and move on to discussing what happens at the PCT. So in the last recordings, the previous recordings, we have talked about the processes that occur when the nephrons produce urine. Okay, so urine production basically involves 3 steps, and that is filtration, reabsorption, and secretion. Filtration happens in the renal corpuscle. Blood is filtered out of the glomerulus into the Bowman's capsule. And now we're on the step where reabsorption occurs. So the proximal convoluted tubule is where the filtrate goes right after it leaves the Bowman's capsule. So what has been filtered is everything. Everything's been filtered out of the blood besides the red blood cells and the large proteins. So we want some of that important material like nutrients back in the blood. We don't want to lose those nutrients because our cells need them. So all of the nutrients like glucose and amino acids are reabsorbed back into the blood. 65% of sodium ions and water are reabsorbed back into the blood. Many ions are reabsorbed, almost all the uric acid and half of the urea. Then we get to the nephron loop. And this is real important that you learn what happens in the descending limb versus what happens in the ascending limb. The descending limb has cells that line the nephron that allow water to leave, but solutes are not able to leave. Solutes are prevented from leaving, so the solutes stay in the filtrate in the descending limb. So as you move down the descending limb, the filtrate becomes more and more concentrated because water's leaving and solutes are not leaving. Then as you go up the ascending limb, it's opposite. Water can't leave, but solutes can. So as you go up the ascending limb toward the distal convoluted tubule, the filtrate becomes more dilute. And then you get to the distal convoluted tubule and the collecting duct. And here we have mostly what's going on as secretion. Any reabsorption that happens as far as materials that are moved from the filtrate back into the blood, this is regulated by hormones. And one of the most important, you may even say the most important hormone that regulates reabsorption at the DCT is called ADH, or anti-diuretic hormone. So to remember what this hormone does, just remind yourself what a diuretic is. We know that there are certain substances that we can consume that cause us to urinate more, to release a larger volume of urine, to go to the bathroom more. They're called diuretics. If you're a coffee drinker or if you drink any caffeine, you know, because when you drink caffeinated beverages, then often that will cause you to have to urinate more frequently, and it'll be a larger volume of urine. So ADH is anti-diuretic. It causes you to retain more fluid, okay? So not to urinate as frequently. ADH is released by the posterior pituitary gland, and it causes the principal cells of the collecting ducts to insert aquaporins in the plasma membranes, and that will increase the reabsorption of water back into the blood. The water will travel through the aquaporins back into the bloodstream. Actually back into the tissue fluid and then into the blood vessels. So increased ADH levels cause an increase in water reabsorption. So you're going to have a lower volume of fluid in your filtrate. Now there's another hormone called aldosterone, and it targets the principal cells of the collecting ducts as well, and the DCT. And the functions of aldosterone are to increase blood pressure and decrease potassium levels. Then we have ANP, or atrial natriuretic peptide, which is released by the atrium of the heart. So it's actually a hormone released by the heart, and it reduces blood sodium levels, and it results in decreased blood volume and blood pressure. When you don't have as much sodium in the blood, remember, water follows salt. So if you reduce sodium in the blood, you reduce water in the blood, and that reduces blood volume. And when you reduce blood volume, you reduce blood pressure. Then we have a hormone called PTH, parathyroid hormone, and that acts on the cells of the DCT to increase calcium reabsorption. So when your blood calcium levels are low, calcium is reabsorbed back into the blood. And this is a summary of tubular reabsorption and secretion. Reabsorption in the PCT we've already covered, and then you can see the descending and the ascending limb of the nephron loop is covered here too, but let's just focus on the DCT and the collecting ducts and look at what happens there. All right, so aldosterone regulates the secretion of potassium ions from the blood back into the filtrate. In the collecting ducts and in the DCT, if you look at the blue box here and then the blue box up here at the top right, the reabsorption of sodium back into the blood is regulated by aldosterone. In the DCT, and it's regulated by aldosterone in the collecting duct as well. And the reabsorption of urea is regulated in the collecting duct, and the reabsorption of calcium is regulated by parathyroid hormone in the DCT. The reabsorption of water is regulated by ADH in the collecting duct, and sodium by aldosterone, just like in the DCT. So the main thing then is to make sure you know the effects of ADH, aldosterone, parathyroid hormone, and ANP. Okay, and then we have the last stage of urine formation, and that is secretion. In secretion, that is the reverse of reabsorption, where reabsorption is materials moving from the filtrate back into the blood. It's like the blood is reclaiming important substances. But secretion is when we're kind of fine-tuning the concentration of the filtrate. And that is when materials are moved from the blood back into the filtrate, secreted back into the filtrate. Almost all secretion occurs in the DCT. The substances that are secreted, they're selected substances, and they're moved from the capillaries into the filtrate, from the blood capillaries into the filtrate. Some of these substances are potassium ions, hydrogen ions, ammonium, creatinine, organic acids, and uric acid, and then certain bases like bicarbonate. Okay, so now we're gonna talk about the regulation of urine concentration and volume. The kidneys produce only small amounts of urine if the body is dehydrated. If the body is overhydrated, the kidneys produce dilute urine. So the kidneys adjust the volume of urine based on, the hydration level, I guess you could say. And this is accomplished through that nephron loop. And the process that occurs in the nephron loop is called countercurrent mechanism. It's called countercurrent. And there are two terms that you need to know that have countercurrent in them. There's countercurrent multiplier and countercurrent exchanger. Countercurrent multiplier refers to the interaction of the filtrate flow in the ascending and descending limbs of the nephron loops. Countercurrent exchanger refers to the blood flow in the vasa recta that, of course, travels parallel to the nephron loops. So countercurrent multiplier is just really referring to what happens in the nephron loop, and it's referring to the interaction of filtrate and the composition of the filtrate in the nephron loop, whereas the countercurrent exchanger is referring to the relationship between the blood and the loop. So the countercurrent multiplier creates a concentration gradient. In other words, it creates the concentrated urine. And the exchanger preserves that concentration gradient. And it is kind of hard to understand. I'm not going to do all this math. But basically what happens, and we'll talk about the ascending limb first, because even though that happens, even though the urine flows down the descending limb and then up the ascending limb, it is what happens in the ascending limb that affects what happens in the descending limb. So the ascending limb of the nephron loop, remember, is the thick portion of the nephron loop. It's impermeable to water and selectively permeable to solutes like sodium and chloride ions. So sodium and chloride ions are reabsorbed in the thick segment of the ascending limb. And water just stays in there because it can't leave since the ascending limb is impermeable to water. So okay, so then I'm sorry, I lost my train of thought. So then in the descending limb, which is permeable to water but not permeable to solutes, water leaves the filtrate, and that causes the remaining filtrate concentration to be much higher. So the combination effects of the ascending and descending limb of the nephron loop is to concentrate the urine. I know it's hard to understand, and I'll try to help you with some visuals here, but you have the descending limb that lets water leave, but the solutes stay. So as the fluid inside gets a darker color, we see it's more concentrated, and the ascending limb is allowing sodium and chloride ions to leave. So the filtrate becomes less concentrated as you go up the ascending limb. But the overall effect is to concentrate the urine. That's the reason for the nephron loop. And so here you can see some numbers that will help you understand this idea of countercurrent multiplier. Okay, so let's look at the ascending limb. I know step one is over here, but I just want you to look at the numbers. Okay, so in the ascending limb, you can see that the concentration is 200 milliosmoles right at this point, okay, up near the top of almost at the entrance of the DCT. So 200 milliosmoles. As the sodium and chloride ions are actively pumped out, and the water stays in, that is going to decrease the concentration as you go up the loop. Okay, but this is the concentration at this level is 200. But over here in the descending limb, the concentration is higher. So what that's doing is you have a higher concentration here that's leading to, if you think about in terms of a concentration gradient, let's even go down to the bottom where there's not so much going on. It's 900 milliosmoles at this level, in the descending limb and 700 in the ascending limb. So this is high, this is low. The 900 is high, the 700 is low. And they're at the same level, so that is going to cause solutes to want to leave. And so in other words, the differences in the fact that the concentration is slightly higher in the descending limb at the same level as it is in the ascending limb, that leads to some of the ions leaving by diffusion. So, and that's the current, what is meant by the countercurrent multiplier is the numbers are higher in the descending limb than the ascending limb. It's really hard to understand, and I'm sorry, you don't have to understand it. You just have to know, like, the only question you would be asked is the nephron loop. The purpose of the nephron loop is to concentrate the urine. It does this by countercurrent multiplier and countercurrent exchange, but that's the purpose of that loop, is to concentrate the urine. That's the reason it's designed the way it is to concentrate, to produce concentrated urine. Okay, so if you remember that, you'll be fine. The only other thing is to know that the descending limb is permeable to water, not solutes, and the ascending is permeable to solutes and not water. Okay, now the countercurrent exchanger involves the vasa recta, and the result of it is the volume of blood at the end of the vasa recta is greater than at the beginning. So it increases the volume of blood. And this is the vasa recta, and you could look at those numbers too, but it's just called a countercurrent exchange. Countercurrent because the current of the blood vessel is opposite. The one that's going down is opposite to the one that's going up. And then it's also opposite to the nephron loop. So the currents are moving in opposite directions. And that's why it's called countercurrent. All right. So I know I've run out of time, and it's making the recording longer, but I want to cover formation of dilute. Well, no, I won't. I'm going to stop here. We will talk about formation of dilute or concentrated urine in the next recording. And this is slide number 40. So we'll stop it. We'll say we stopped at slide 39.